imagine a similar situation for cancer cells. So the green line is showing what would happen to a normal cell if you reduce the HSP90. You'll get to a point where it's going to have an effect because HSP90 is essential for growth. But normal cells don't need all of the HSP90 that they have. In normal cells and tissues, HSP90 makes up somewhere around 3% of the total protein of the cell. So it's quite a lot. It's in micromolar concentration in cells. And they can get by with a lot less. But if you stress the cell exposure, which has a significant effect. You get lots more cell death. So far, the data suggests, and additional information that has come out since this support the fact that indeed you can affect the robustness of cancer cells with relatively low dose HSP90 inhibitors. So how do HSP90 inhibitors inhibit HSP90? So it was unclear when we got into this and we identified the first small molecule inhibitor of HSP90, which is a version of 17-AAG, in 1994. It was unclear whether HSP90 was a chaperone that required ATP or not. There are examples of both kinds of chaperones. Using these small molecule inhibitors, it's become very clear that ATP binding and hydrolysis are essential for HSP90 function. I'll show you why. As it happens, these first inhibitors of HSP90 that were identified, 17-AG is a derivative of galvanamycin, and then a second structurally distinct. Those are both right. This is the end-domain ATP binding pocket of HSP90, and you can see ATP bound here on the left. And to everybody's surprise, these small molecule inhibitors bind in the ATP binding pocket. So one reason why it was controversial up until this point whether HSP90 bound ATP, required ATP, etc., is that its binding affinity for ATP is extremely poor, and its ATP hydrolyzing activity is extremely poor. But with these drugs, we can now specifically measure HSP90 dependent ATPase activity because only the ATP bound to HSP90 is going to be affected. So contaminating proteins in preparations like kinases, etc., with much higher ATPase activity are not going to be inhibited by these drugs. And you can see the very small amount of ATPase activity coming from HSP90 is fully inhibited by these agents. So HSP90 exists as a dimer. The dimerization domain is in the C-terminal part of the protein. ATP binds to the N-terminal part, shown in blue. And in the presence of ATP, HSP90 undergoes a series of conformational changes, which is important for ATP hydrolysis and for chaperoning of clients. In the presence of these HSP90 inhibitors, however, ATP can't bind because the inhibitors have much higher affinity for this binding site than does the nucleotide. And HSP90 remains in this inactive conformation, which leads to the degradation of any client proteins bound to HSP90 via the proteasome. And we had identified two E3 ligases that ubiquitinate proteins so that they are recognized and degraded by the proteasome. Both CHIP and Cullen-5 actually associate with HSP90 and are responsible for ubiquitinating clients that are not allowed to proceed through a chaperone cycle because HSP90 inhibitors are blocking it. So development of HSP90 inhibitors picked up quite dramatically after we reported the first one in 1994. And you can just see here over the last 25 years or so how many HSP90 inhibitors have been developed. And all of the ones shown here have been evaluated in clinical trials. The little red top to the panel on the right-hand side is a special case of an HSP90 inhibitor that I'll get to in the last part of the talk. But suffice it to say that a lot of HSP90 inhibitors have gone into clinical trials. And yet no inhibitor has yet been approved by the FDA as having any single agent activity. And these, unfortunately, are the few HSP90 inhibitors that remain in clinical trial as of August of this year. So nowhere near as many as were shown on the previous slide. And a couple of these trials are simply trials for imaging of HSP90 with iodinated versions of an HSP90 inhibitor. But there still remain a few phase 1 and phase 2 trials that are actively recruiting. These combination trials, which most of these on this table are in combination with previously approved drugs that target this particular kind of cancer. The indication is shown on the right of the slide. And so you can see that now most of the clinical trials are not so much focusing on HSP90 as the single agent, which we would have suggested from the beginning was not the way to develop this drug. It's best used in combination because what the radiation experiment showed a long time ago was that we can affect the robustness of a cell exposed to something else when we put in an HSP90 inhibitor. So a lot of the agents used in combination are kinases, but not all of them are. These trials are open, and it's hopeful that we'll see some interesting results. These are primarily phase 2s. There are some phase 1s also included. So about 6 years ago, interest was increased that perhaps we could develop an HSP90 inhibitor for clinical use, and in part based on this study in HER2-positive breast cancers, where patients who had been treated with the antibody to HER2, Herceptin, initially respond and then stop responding, and so everybody was progressing on the antibody alone. Now, HER2 turns out to be a kinase, and it's very sensitive to HSP90 inhibitors, very dependent on HSP90. And when these people added in 17-AAG to Herceptin, which, remember, was no longer working in these patients, there was a 63% increase in benefit and a 26% response rate, which is actually quite high. So people got all excited that combination with a HER2, with an antibody that targets HER2 may be a great use for HSP90 inhibitors, and this is still under investigation even now with newer, more synthetic HSP90 inhibitors and with different antibodies to HER2. Some other interesting data that study in non-small cell lung cancer with gantenerumab, which is a more recent HSP90 inhibitor, in patients that are positive for ALK. ALK is a kinase that is frequently mutated in a reasonable percentage of lung cancer patients. And all those green the HSP90 inhibitor, you see this patient, first of all, has stage 4 lung cancer, which is very advanced, received crizotinib, which is an ALK inhibitor for 1 year, and progressed on that, and then enrolled in this trial looking at canetespib, and you can see that these tumors, which were no longer responsive to an ALK inhibitor, nonetheless are shrinking after 3 doses of the HSP90 inhibitor. So there are trials underway right now combining ALK inhibitors with HSP90 inhibitors as opposed to just looking at HSP90 as a target after the patient stops responding to the ALK inhibitor. Here's another example in a lung cancer with a BRAF mutation. BRAF is also a minus of HSP90. This patient received a lot of chemotherapy as well as targeted agents, including a proteasome inhibitor, and at this point was not responding to anything. But after 4 cycles of HSP90 inhibitors, this lesion went from this size to this size, and the duration was for more than a year. So there are definitely hints of activity, even single agent activity, but we're asking these HSP90 inhibitors to now cause regression of tumors that have stopped responding to everything else that's been tried, and after they develop resistance to each of these treatments, they get more and more aggressive, and this is a very high bar for any agent to cross, including an HSP90 inhibitor. This is a triple-negative breast cancer, again receiving a lot of treatment and showing what happens after 3.5 months, after 4 months of benitetazine. So, there's a lot of anecdotal data suggesting that HSP90 inhibitors should work in cancer. Why haven't any been approved yet? We started looking at other things that HSP90 inhibitors might do that might be counteractive of their anti-tumor activity. And one of the things that we and others have found is that HSP90 inhibitors seem to potentiate the heat shock response by activating the master transcription factor of the heat shock response, HSF1, which is activated by the same kinds of stresses that HSP90 looks for, and that's because HSP90 itself is the transcriptional target of HSF1, as is another molecular chaperone, HSP7. So what we're showing here in a Western blot in a cell line is that increasing concentrations of galvanomycin cause the degradation of ERB2, also known as HER2, which is that kinase that's very sensitive. But at the same time, they cause an increase in HSP70, which is indicative of this induction of the heat shock response mediated by HSF1. And you'll notice it's only the heat-inducible HSP70 in the red box at the bottom, not the constitutively expressed HSC70, whose levels are going up, suggesting this really is a transcriptional effect that's mediated by HSF1. And if you look in patients receiving an HSP90 inhibitor, you can look at HSP70 mRNA induction in lymphocytes of patients, and you can see from the data shown here that there is a correlation that's quite strong between the dose of HSP90 inhibitor given and the increase in HSP70 mRNA. And the higher the dose and the more cycles of treatment, the bigger the increase in HSP70. And this is plotted in the 2 graphs on the bottom, and you can see that the correlation is quite remarkable. So this isn't just a phenomenon of cell lines in vitro, it's happening in patients as well. A number of years ago, the Lindquist Lab published a paper reporting that not only does HSF1 drive genes associated with the stress response, they also have a program of cancer-specific gene expression. And so you might think that if you're using an HSP90 inhibitor, which is upregulating HSF1 in some fashion, or at least upregulating its activity, if this leads to upregulation of this cancer-specific gene expression, you're going to be shooting yourself in the foot in terms of using this as an anti-cancer treatment. This lab also looked at survival with breast cancer. Now, instead of stratifying for HSP90, you stratify for HSF1, and they found again that the high-expressing HSF1 samples in those tumors, the patient's survival was negatively affected. So again, there's a negative effect of high HSF1 in tumors, just like there is of high HSP9. And if you look across a number of cancers, if you just look at gene expression at the mRNA level, the types of cancers are shown on the x-axis, and it doesn't really matter which ones they are. But you can see in the red bars is HSF1 expression, and the green bars is c-Myc expression. c-Myc is generally considered to be a paradigm of a cancer-related transcription factor that's frequently overexpressed in cancer. And you can see that HSF1 is in fact far more overexpressed as a rule than is c-Myc. So cancers need HSF1. If your anti-cancer drug is in any way activating HSF1, that's not going to ultimately be a good thing. So for a long time, the model of how HSP90 is related to HSF1 is that monomers of HSF1 are sequestered by HSP90 until there's a stress. And when there's a stress and you get misfolded proteins, HSP90 comes off of the HSF1, goes to deal with the misfolded proteins, and these monomers of HSF1, which are not active until they trimerize, are allowed to trimerize and become active transcription factors. That's been a model that's been around since the mid-90s. So if you take this and say, okay, what do HSP90 inhibitors do? The speculation was the HSP90 inhibitors act like another cell stress. So there's an HSP90 inhibitor, binds to HSP90, and then the promoters are allowed to trimerize and become active. So this was the model. HSP90 functions as a repressor of HSF1. It functions as a sensor for proteotoxic stress, and release of HSF1 from HSP90 is the key event in induction of the heat shock response that's driven by the transcription factor. So we asked a number of years ago if this actually makes sense given the data that's accumulated over the last 20 years. And the answer is no, it doesn't make sense. First of all, endogenous interactions between wild-type HSP90 and HSF1 are almost impossible to visualize without cross-linking. So that doesn't suggest that HSP90 is sequestering HSF1 in the cytoplasm, or it would be much easier to see these interactions. And a recent study done in vitro with purified proteins, in fact, suggested if you add HSP90 beta, which is one of the two isoforms of HSP90, to HSF1, it promotes the temperature-dependent trimerization and activation of HSF1. So exactly opposite to what the old model had said. So we re-asked the question, can we visualize stable interaction of HSP90 with HSF1? And to do this, we use two well-characterized point mutants of HSP90. The one on the left, D93 in HSP90 alpha and D88 in beta, they can't bind ATP, and so they remain in what's called an open conformation where the endomains are undimerized. This basically is a non-functional HSP90. But it's a conformation that at ground state is the most common in a cell. The mutant on the right, E47A in alpha and E42A in beta, can bind HSP90, and so the endomains come together, but they can't undergo ATP hydrolysis. And so this endomain dimerized conformation essentially is stabilized. As you might expect, since this is a conformation that is on the way from open to ATP hydrolysis, is a very unstable conformation without this mutation. It's transient, and it's very hard to identify in human HSP90. So when we look for interaction of either wild-type HSP90 or these two mutants with HSF1, we found that only in the case of the E47A and, to a lesser extent, E42A with beta, you could see robust interaction of HSF1 shown in the top plot there when you pull down flag-tagged HSP90. So HSF1 is indeed able to bind to that ATP-bound closed trap conformation, whereas we could see no interaction of wild type or of the open mutant. So this got us to thinking a little. If the ATP-dependent closed conformation is a transient state and only infrequently accessed by the chaperone, what does this mean for what HSP90 is doing to HSF1? We next asked if HSP90 inhibitors have any effect on this interaction. And as you can see on the left-hand group of panels there, these three HSP90 inhibitors, which have all been in the clinic, all dissociate HSF1 quite efficiently from HSP90 and very quickly, whereas two other inducers of the heat shock response and two other activators of HSF1, heat shock itself, shown on the right, and a proteasome inhibitor right next to heat shock, don't cause dissociation of HSF1 from HSP90. So what this says is that this ability of N-terminal HSP90 inhibitors to dissociate this complex is unique, but it is not required for activation of HSF1. So that again goes counter to the model that's been around for quite some time. So we did a reporter experiment based on including the promoter region of HSP70 to which HSF1 binds, coupled to luciferase. So we wanted to see the effect of 17-AAG, heat shock, and the combination on HSP70 promoter-dependent luciferase activity. And what you can see here is that 17-AAG in this transiently transfected model has essentially no ability to induce reporter activity. But compared to heat shock alone, 17-AAG has a combinatorial effect when added to heat shock.